Hi, we're the Wang Lab, and we will be presenting our work in developing protein markers for quantitative Western blot analysis. My name is Vaishnavi Konda, and I'm a senior at Irvington High School.
My name's An. I am a senior at Gunn High School.
My name is Ayana. I'm a senior at Los Altos High School.
My name is Mark. I am a junior at Los Altos High School.
My name is Aman. I'm a senior at Dublin High School.
My name is Eben. I'm a senior at Lee High School.
My name is Iris. I'm a junior at Design Tech High School.
My name's Andy. I'm a sophomore at Mission San Jose High School.
My name's Anand. I'm a senior at University High School.
Okay. And so we're going to go into, this is just our agenda for what we're going to be going over for today. So first we're going to be going over some background, basically what a protein is and what a Western blot is. And then we're going to go deeper into the Western blot technique. So what's the procedure for a Western blot, the steps and why people do Western blots. And then after that, we'll be going into results and analysis. So how do researchers analyze Western blots, and also what are our current progress on developing quantitative Western blot quantitative markers.
So first, a brief overview of proteins, which are large amino acid polymers. There are 4 levels of protein structure: the primary, secondary, tertiary, and quaternary levels. At the primary level, proteins are chains of amino acids. At the secondary level, proteins take on one of 2 structures, either an alpha helix or beta-pleated sheet. Proteins at the 3rd level are 3D structures, and finally, quaternary level proteins are comprised of smaller protein subunits. Proteins have a variety of functions within organisms, including catalyzing metabolic reactions, being involved in DNA replication, maintaining homeostasis, and much more. They differ from one another in their sequence of amino acids, which results in specific protein folding that then determines its activity.
So what is the Western blot technique? Western blots are widely used for protein expression detection. First, cellular proteins are prepared, then the proteins are denatured and reduced to obtain their primary structure. Coating with SDS imparts a relative negative charge proportional to the molecular weight of molecules, allowing the separation of protein by size through gel electrophoresis. The proteins are then transferred to a nitrocellulose membrane, washed, and then blocked to prevent nonspecific binding before primary antibodies are added. The membrane is then washed again before adding secondary antibodies, which act as a signal for luminescence in order to visualize the amount of protein.
So the first step of the Western blot process is the SDS phase, which is a form of gel electrophoresis. So in this process, a polyacrylamide gel is placed into an apparatus filled with SDS buffer. So SDS denatures proteins, unfolding them into their primary linear structures. SDS also coats the proteins with a negative charge, allowing them to move down the gel from anode to cathode, separated based by size. Larger proteins tend to take longer to pass through the gel. Marker proteins, such as the protein listed in well 5 of this diagram, are used as a reference to determine the molecular weight of other proteins in the gel. The next step of the process is the transfer. In this process, using a biorad transfer apparatus, the gel with the expressed proteins is placed on top of a nitrocellulose blotting membrane. The gel and the membrane are then sandwiched between filter paper and placed into electrode cassettes. These cassettes are then placed into a transfer apparatus filled with buffer to facilitate the flow of electricity. An electric current is then run through this apparatus to transfer the proteins from the gel onto the membrane for further blotting and analysis.
So first, the membrane is blocked with PBST and milk. And this is to prevent non-specific binding, which is when the antibodies bind to unwanted proteins. Next, the primary antibodies are added to the membrane with PBST and milk, and they're incubated. And these antibodies will bind to the proteins on the membrane. Then they're washed. This is so that unbound antibodies can be removed from the membrane. Then secondary antibodies are added to the membrane with PBST, and then they're incubated. This will add and amplify chemiluminescence through the horseradish peroxidase enzyme, or HRP. This will finally allow us to analyze the results through imaging. On the right-hand side, one can see fluorescence secondary antibody technique, and in this, a substrate like HRP is not required.
Western blot imaging consists of first capturing an image of a membrane after the protein separation and transfer process. The image is then used to visualize protein bands, which can then be analyzed to determine protein quantity as well as molecular weight. The red column to the left represents the protein marker, which encompasses different known protein molecular weights. This is then compared with the protein of interest and also functions as a monitor of the separation process that the gel runs in the gel electrophoresis. The imaging technique used here is fluorescence imaging, with the green fluorescent protein being the protein of interest.
Now, at this point, you're probably wondering, why use Western blot? Well, Western blots have gained popularity amongst researchers due to various reasons. The first of these being their versatility. Western blot can detect and quantify specific proteins from complex mixtures, making them very adaptable to various research fields involving protein development and protein analysis. Also, another reason why they're popular is that their ease of use. While requiring some technical expertise and familiarity with various instruments, Western blots have a widely established protocol and they're very accessible. Lastly, the imaging of the Western blot allows for us to have a visual representation of protein bands, allowing for straightforward qualitative and semi-quantitative analysis. And due to all these reasons, Western blots have been the most commonly used technique when it comes to protein analysis in recent years. And as stated by the Journal of Proteomics, Western blots are included in 8 to 9% of all protein-related publications from the last 30 years.
So the first step is to isolate a plasmid from bacteria through a process called mini-prep. This plasmid will contain the marker of the marker proteins that our lab is developing. The next step is to test the expression of that plasmid by transforming the plasmid into bacteria such as E. coli. Then the next step is to purify these proteins from the bacteria. And lastly, we analyze and quantify the proteins using techniques such as SDS, page, and Western blot, and finally image.
Quantification of the amount of protein is typically done through image analysis of the membrane using a software called ImageJ. After selecting each lane in the gel, which are the vertical strips on the image on the right, the software shows the intensity of each band in the membrane as a graph. And we take the area of each of these graphs to find the total intensity of each band and compare these values to a standard set of bands for which the protein concentration is known. And the standard has traditionally been various housekeeping proteins.
So to add on to that, housekeeping proteins are proteins that are assumed to have consistent expression across different cell and tissue types. For example, a very popular one is GAPDH. It's used in glycolysis, DNA repair, apoptosis, so it's very common amongst all cell types. And so it's used as like a baseline to compare changes in target protein expression. And so how researchers will typically use housekeeping proteins to assess changes in protein expression. They'll first quantify target protein intensity using the ImageJ analysis, for example, and also they will quantify housekeeping protein intensity. And using these numbers, they will normalize intensities of the target protein against the housekeeping proteins.
So one of the most significant issues with Western blotting is signal oversaturation. So signal oversaturation occurs when the detection signal is too strong and leads to bands that appear uniformly intense, regardless of the actual protein quantification and inaccurate. Another challenge is the use of nonlinear controls, which are reference bands that do not increase linearly with protein concentration. Nonlinear controls can lead to errors in interpreting the relationship between band intensity and protein quantity. Furthermore, consistent and transparent findings are essential for reproducibility. However, it's common to see short citations in publications where only a general reference is provided instead of a detailed protocol. Also, limited information on the antibodies used in the experiment, including their sources and dilutions, can lead to inconsistencies in experimental outcomes. And finally, Blot cropping and non-disclosure of raw blot data can lead to exclusion of important bands or controls, which can skew interpretation of results and make it difficult to assess the integrity of the findings. And it's also important to consider that Western blots are able to measure increased or decreased protein expression, which is one of the most measured biological events for biological changes. So Western blot remains the only mainstream method for observing this protein modification. So it's essential to optimize this process and fix the quantification issues.
Now, here are some examples of these issues, starting with nonlinear controls. And as mentioned previously, during the Western blot analysis process, the ratio between band intensity of the known concentrations of housekeeping proteins is used to calculate the concentrations, as well as their intensity is used to calculate the concentrations of the unknown samples. Therefore, this ratio needs to be consistent in order to provide an accurate measurement for a range of concentrations. However, the most commonly used housekeeping proteins, such as GPDH, alpha tubulin, and beta tubulin, are often used outside their linear range, resulting in discrepancy during quantification. And as you can see on the figure on the right, the concentrations of GPDH and beta tubulin do not have a linear relationship with their intensities, meaning they cannot be used as a valid benchmark to quantify unknown concentrations. And this ties with another issue of protein oversaturation, which occurs when the amount of protein has reached a point where it goes over the membrane capacity, and not all proteins are able to be detected, since the binding antibodies can only bind to the proteins on the top layer of the protein stack. Since not all proteins can be detected, this results in underestimation during the quantification process. And this can also lead to signal oversaturation, where the intensity of the band is simply too strong for an imaging software like ImageJ to accurately analyze. And this can be seen in Figure D with the bands of GPDH, and they all appear to be similar, but in reality, they contain very different amounts of protein. And this type of issue is most commonly seen in Western blots that use chemiluminescent, but it's also possible to see this issue in Western blots that use fluorescent detection. And in both cases, oversaturation leads to inaccurate results because after a certain point, the Western blot no longer accurately reflects the amount of protein that you have.
To address these issues, we have developed and are currently testing a set of protein markers based on GAPDH that we believe show a clearer linear progression of intensity as the protein concentration increases. Figure A shows the design of 3 different protein markers, each with a histone 6, c-MYC, HA, and flag epitope tag at the N terminal, which are amino acid sequences that primary antibodies can bind to. The GAPDH markers have the molecular weights of 22, 44, and 86 kilodaltons, and in order to achieve this, marker 3 has a truncated GAPDH protein. The protein markers were expressed and purified from E. coli, then SDS page and Kumasi staining were performed for the combined markers together, as seen in Figure B. And because of the differences in molecular weights, the 3 distinct bands appear even when combined into the same well. In the Western blot shown, each marker is around 0.5 micrograms.
Here in Figure A, we have quantified JPDH protein markers using the Western blot. In lane 1, different amounts of JPDH were loaded, which are 1, 0.5, and 0.25 picomoles from top to bottom. And in lane 2, a constant amount of 0.5 picomole GAPDH sample was loaded. In Figure B, the intensity of each marker on lane 1 was measured using ImageJ software and was plotted in a standard curve against the input amount. Finally, in Figure C, the intensity of each marker on lane 2 was also obtained, and the correlating amount of protein was calculated for each marker.
So here's an example of an application of Western blot, where we can accurately quantify cellular protein product in a standardized manner. On the left is a Western blot of HEC293 cell lysate, in which we are looking for our target protein, the programmed cell death ligand, or PDL1. PDL1 is a protein that acts as a brake to control the body's immune response. It inhibits T cell activity, so many cancerous cells overexpress the protein to avoid being detected by the immune system. The HEC293 cells were collected and then lysed or broken down using REPA buffer. Regarding quantitative markers, there were 3 different sizes of markers that were run as seen in the first 3 lanes of the gel. Our PDL1 is recombinant PDL1. It's engineered, and it's seen in the rightmost lanes of the membrane. Our PDL1 serves as a positive control. If there is PDL1 in our HEC293 cells, the bands will be the same as the RPDL1 bands. After running the gel and completing the transfer, we use the chemiluminescence antibody detection method previously mentioned. As seen in the Western blot, there are no bands present in the HEC293 lysate lanes and bands present in the RPDL1 lanes. Upon further quantification using ImageJ software and the creation of a standard curve, which is seen on the right, we can determine that RPDL1 was loaded at a range of around 10 to 15 nanograms and that there is no PDL1 expressed in the cells. Quantification, therefore, provides more detailed insight into biological events like cell health and can provide us with numerical data. This then leads to higher quality research and reproducibility. The optimization of Western blot is highly useful for analyzing biological changes.
And in terms of our future steps, we're just going to continue working on our set of quantitative markers for analysis and finalizing it so that we can achieve absolute quantification, and also continue testing on real samples for quantification of proteins just as further proof of.
This is the second of three presentations.
Finally, we would like to extend a warm thank you to Dr. Zhong Wang for his continuous guidance and support and mentorship during our research process, and as well as a big thank you to ASDRP for providing us with the equipment, training, and laboratory space for our research. Thank you.